Welcome back to our online learning method. In the last lecture, we looked at the life cycle of a flowering plant or an angiosperm. What was important there, or what was rather new, was the development of the embryo sac. In everything that we did, the important thing was that we identified the egg in the embryo sac, and we identified in the pollen the sperms. When these two come together in a process we call fertilization, we then get the embryo, and in a process we call double fertilization, the other sperm fertilizes the polar nucleus to give us the endosperm, or the food of the embryo. The embryo, once formed, doesn't just grow haphazardly. It grows in a particular sequence. The area of growth of the embryo is called a meristem. The word meristem, as presented here, comes from the Greek word meristos, that means dividing or divisible. Meristems are areas of active division. Division not just for fun, but to add more and more cells so that the embryo becomes bigger and bigger. Now, meristems are identified by the following. The cells in the meristem will be actively dividing. And if they're actively dividing, please note that there will be small or no vacuoles. The important thing is that these cells have a large nucleus. These cells are young, and therefore their walls are thin. Please, the fact that I said cells are living is not a stupid idea. Not all cells are alive. So when you describe a cell, it is important to say whether a cell is living or if it is dead. Some cells mature so that they can die. These cells mature and remain alive. Because these cells are actively dividing, there are no spaces between them. Hence, these characteristics define a meristem. Meristems can be classified in a number of ways depending on what you want to address. The stage of development means that you are saying whether that meristem is a meristem of the embryo or it is a meristem of a mature plant. Promeristems are areas of initiation of new cells, especially in embryos. However, in a growing plant, you can say this is a primary meristem or a secondary meristem, meaning that these are growth areas in a young plant, or these are growth areas in an old plant. The second way we can say is the position in the plant. If we say these are apical meristems, we mean they're either in the shoot or in the root, in the opposite or in the growing tips. Intercalary means these are meristems that are located anywhere else in the growing plant. Lateral, as the word says, it is on the side. These are the meristems that will give you the branches. Intercalary, they will give you the height. Apical, they supply the initial tissue. We can look at meristems by their function. Function here means what they are eventually going to produce. The meristem that eventually gives us the epidermis is the protoderm. The meristems that gives us a vascular cylinder is a procambium. Please note, this cambium here does not refer to the cambium in the vascular cylinder. It is just a terminology for the meristem. The meristem that gives you the cortex or the pith is called a fundamental or a ground meristem. So this just tells you what the meristem is going to end up giving you. Now, meristems function in a particular order. The cell that initially divides is called the initial. If you have a cell and it divides into two, those two cells are exactly the same, but we call one an initial and we call the other one a derivative. That means we are assuming this one will go up, will grow out of the meristem and produce something. This initial will stay in the meristem and give us more cells. But please note, there is no difference between these two cells. It is just a terminology we give. Therefore, a derivative can become an initial, and an initial can become a derivative. A derivative. Okay. I've already explained these meristems to you, and these meristems are meristems that you will find in the embryo, embryonic tissue. These meristems, which are going to give you the epidermis, the ground epidermis, and the vascular system, become apparent by the shape of the cells, the location of the cells, and the overall function that they will eventually produce. You cannot expect a cell that is on the outside to be a procambium. A procambium will be somewhere in the center of the cell and it will be elongated. A ground meristem is a cell that is often more rounded. Protoderm or dermatogen, these are cells that become elongated along the periphery. The divisions these cells go through are said to be periclinal if the division is parallel to the periphery, anticlinal if the division is at right angles to the periphery. Periclinal and anticlinal divisions occur all over the plant. Okay. Now, when you look at a cell, the important thing, you should look at its location. That gives you an idea what the cell should be doing. And when you describe a cell, I've already told you that vacuolation tells you exactly how far away from the meristem the cell is. Closer to the meristem, the cell will be less vacuolated, and further away from the meristem, the cell will be more vacuolated. So what I was telling you here is that the cells of the protoderm are more elongated because they have to fit along a broad area. The cells of the procambium are elongated. The cells of the ground tissue are more isodiametric. Isodiametric means the sides are almost of the same size. Apical meristems, as I've already told you, can be in the shoot or they can be in the root. Details are already given to you here, but please note there are no spaces between cells. Cells are contiguous. Contiguous is a terminology we use when we mean kind of continuous. Okay, now let us look at apical meristems. There are two apical meristems, that of the shoot and that of the root. These are not the same. The shoot apical meristem is complex. It's complex because it has to take into account the formation of leaves, which we call leaf primordia. Primordia means the primary meristem that will give rise to the leaf. The swelling, the mound, is a primordia. The root apical meristem, by comparison, is simple because everything just goes from one center to produce the rest. The only complication or the only difference is the fact that it has a root cap, which you will not find in a shoot apical meristem. Now, I said cells are going to be what they are depending on where they find themselves. Consider this: the pro-meristem, the region of initiation. Cells that find themselves in the periphery are going to be elongated and be part of the protoderm, continue to give us the epidermis. Cells in the center will be elongated and they will give us the cells of the vascular system. Cells in between are more like cushioning cells. They are very similar, and these cells are thin-walled and isodiametric. They are a living joining between the center and the outside and the center. This is very important to be aware that cells talk to each other. No cell exists in isolation. In your books, you will find these diagrams. I've twisted them like this so that they could fit, but this is the apical meristem. This is the shoot apical meristem, showing you this apical shoot, and this lateral shoot is the leaf primordium. We realize that the cells in the meristems are actively growing. You can see that they're very dense together, and as we get away from the meristems, individuals become clearer. And as we get away from the meristem, we realize which cells are in the periphery, which cells are in the center, and which cells are in the middle. As you can see, there's a lot of confusion here because there are these leaves that protect the apical meristem. It's a sensitive area, it shouldn't be exposed, so it is protected by these leaf primordia as it grows out. This is the root apical meristem. I will come back to it, but please just note the following, that there is an area here. There's an area here which we can demarcate as the source, and from this source things are growing up and things are growing down. From this source we can see a central unity here, and from this source we can see the periphery over here, and then in between we see these groups. Now these cells in between are the ground meristem, and in here we have the apical meristem, and in here we are going to have the procambium, which is going to give rise to the vascular system. Now, remember that when we started studying the plants, I told you that we are following a sequence which we believe is the way in which plants evolved. We came from bryophytes to pteridophytes to gymnosperms to angiosperms. Now, when we look at the inside of the plant, the topic is called anatomy. When we are looking at the cell arrangement, even the cell arrangement differs from one group of plants to the next. We believe that ancient plants were much simpler than the plants we have now. And here we have Equisetum, which is an example of a pteridophyte. In Equisetum, we can see that the cells here seem to be coming from this cell. The cells that come out here, if this were your pyramidal bread, you slice it like that, you get these cells. You slice it like that, you get these cells. You slice it like that, you get these cells. So in Equisetum, we can identify an individual cell, a single large initial. This is common in pteridophytes. However, in gymnosperms are the naked-seeded plants. Instead of a single cell, we observe a group of cells, a region. Here you can see individual cells, and we can see that the nucleus here is prominent. So here we don't have a single cell, we have a group of cells. There is a difference between gymnosperms and angiosperms in the way in which the apical meristem is arranged. We see this group, and then we see there is a cover here, and there is another layer here. But if we look at a gymnosperm, we only see one layer here, whereas here we can see two layers. This is Pisum, the P, which is an angiosperm. We see two layers. In Pinus, we only see one layer. There was a scientist who studied this apical meristem and realized these differences. We will go through the people who have contributed to these studies one at a time. Okay, so the arrangement of the meristem was studied by a number of people, and way back in 1858, a scientist by the name of Nageli, I've given you the reference in the book you're supposed to have, he came up with the apical cell theory. Please note, theories are true for certain individuals. It doesn't mean because they're not true for everything that we reject them. This theory said there is a single solitary cell in the apical meristem. This was found to be true in cryptogams, which we have already covered, the algae, the bryophytes, and the pteridophytes. But it is not true for phanerogams or seed plants. That means angiosperms and gymnosperms. So there was a need to come up with another theory that will explain what happens in seed plants. A number of theories came up. One individual by the name of Hanston in 1870 came up with a histogen theory, and another individual in 1924 by the name of Schmidt came up with another theory which we call the Tunica corpus theory. Let us go to the histogen theory first. The histogen theory, the histogen theory says in the meristem we are going to observe the histogen. The histogen is the area of active cell division. Now, in the histogen, we are going to observe a dermatogen, a periblem, and a plerome. The dermatogen in the stem will give us the epidermis, the periblem will give us the cortex, the plerome will give us the vascular system. And in the root, the dermatogen will give us the epidermis as well as the root cap or the calyptrogen. The rest are true, except please note, in the root, the vascular system is called the stele. In the stem, it is called the vascular bundles. Okay. The details of this are also given in your book here. Now, let us go to what Schmidt said. Schmidt presented what we call the Tunica corpus theory. Allow me to go back to the pictures I presented to you. Okay. The Tunica corpus theory, a tunic, for ladies, they know that a tunic is a dress. A tunic is something you wear on top. So the outside covering is the tunica. Here we have a one layer tunica, and here we have a two layer tunica. Now this mass of cells inside, the body of cells, is called corpus. For us Catholics, when we receive the body of Christ, we say corpus Christi, meaning the body of Christ. So there is a body and there is a dress. Corpus and tunica. So these illustrate the corpus tunica theory of Schmidt. Okay. All I've said is explained to you here in notes. I assume that now that I've spoken to you, you'll be able to read this and be able to follow. The only thing I want to emphasize is the fact that it is possible to use anatomy to portray or to deduce about evolution. So somebody did work to look at anatomy and evolution, and this was done by a fellow called Voronin. You'll find it somewhere in these notes, and he explained how, here it is over here, Voronin is the one who looked at phylogenetic relationships of plants by looking at their anatomy. I've already presented this to you in the sense that I said to you the Nageli single cell apical meristem theory will occur in primitive plants. The single tunica in the next row of plants, that means the gymnosperms, and the bitunicate plants are the current or the present day flowering plants which we have. Okay, now this is explaining something in the root. We have said phylogeny or evolution can be explained with the tunica corpus theory. Please note the tunica corpus theory applies only in the shoot apical meristem. In the root apical meristem, we have other theories, but I'll present to you this one. And this theory is also asking how many initials are in the meristem. That means in where the cells are growing, are they coming out from one cell, or are they coming out from a group of cells or from a kind of organization? We observe two types of theories. The first theory, which I've called type 2, I'm starting with it because it goes to the single initial in the meristem. Now, if we had sectioned a growing root, we wouldn't see this kind of diagram. This diagram is an interpretation of this, which is what you actually see. What we are looking at here, it says we can observe these cells here, which are different from these cells here, which are different from the cells on the outside. However, all of these cells seem to be coming from somewhere here. Let us look at this individual cell, and we can see how this individual cell can produce all these cells by anticlinal and periclinal divisions. Now, this is the interpretation. This interpretation, which was also observed by Voronin, says there is a single initial, and from this initial we get the vascular system, we get the cortex. From this initial we also get the epidermis. From the same initial we are going to get the root cap. The root, all the cells of the root will come from this. This was observed to be true in certain plants. Please note, this in the root was observed in spruce, which is a gymnosperm. Nageli observed the single initial in the shoot in pteridophytes. So the rate at which the root and the shoot develop is not the same. Now, what is the second observation? The second observation says there are three initials here. In this area here, we are going to observe three initial. One initial here that will give rise to that, another initial that will give rise to this, and another initial that will give rise to the root cap. Okay, these, only when we are a trained anatomist can you interpret this. However, we can all see clearly on here. What this is saying is that there are three initials. You can't see them here, so I've taken them out here. The first initial gives rise to the vascular cylinder, which is this. The second initial gives rise to the cortex, which is here. And the third initial gives rise to both the epidermis and the root cap. This was found to be true in grasses or monocots. Okay? The difference between that one and this one, both of them say that three initials. The difference comes with the third initial. Let me say the second and the third initial. Here we said the second initial will give rise to the cortex and the epidermis. Here we are saying the epidermis is coming from the same initial that gave rise to the root cap. So the third initial gives rise to the root cap and the epidermis. This was observed in dicots. What does this all mean? This all means that the arrangement of meristems has a phylogenetic component, and it is not the same in all plants. Monocots have a different arrangement, dicots have a different arrangement. As to which is older, the monocot or the dicot, taxonomists do not agree. Others believe monocots are more improved, others believe dicots are more improved. What the important thing to note here is that there is a difference in the meristem arrangement in the root. Okay, everything I have said is explained in all these pages except what I'm about to tell you now. The fact that there is, okay, if we're done our practicals, we would realize that in the root, in the stele, we have a special group of cells which we call endothermal cells. Remember, you met these cells in pine in the leaf. They are the same cells. They are characterized by the Casparian strip, a strip made of suberin, which is water resistant. Why should we have this strip? This strip is to force water not to go in between cells, but to go through cells. Okay. However, there are exceptions where you find that the endodermis doesn't have the Casparian strip. Now, those cells are then called passage cells. They allow free movement of water. There is a need for this sometimes as part of the adaptation of the plant for certain situations. Sometimes you want the water to pass quickly or want the water to go through slowly. This is a transversal section of the root, and it's not the whole of it. We haven't shown the epidermis. We have shown you part of the cortex. The important thing is to show you the location of the endodermis. The endodermis is the last layer of the cortex. Just below the endodermis, you have parenchyma cells. These are parenchyma cells. One row of parenchyma cells, which we call the pericycle, before you get to the stele. Now, the stele is mainly the xylem, which I've shown you here in red, and the phloem, which I've shown you here in green. But please know that in the stele, we also have parenchyma and sclerenchyma, which I will explain to you in the next lecture. Okay, so in a practical, we should have looked at the TS of a stem. Please look at that. Okay. Let me just do this. Before I go, here I've tried to show you the section which you would have seen in the root. The root, of course, has the root hairs, which are an extension of an epidermal cell, the cortex, and these dots here are showing you the endodermis, then the pericycle, then the phloem, and the xylem. And of course, if it is a growing plant, it will have a vascular cambium. Okay. Now, I think at high school you were presented with this, and you were told that the root has these meristems, the protoderm, ground meristem, the procambium, and this area which they called the quiescent region, this quiescent region is the promeristem. Okay, I think now we are all at the same page, and as the root grows, the cells that are produced, when the cells are produced, they're more or less the same, but they differentiate. That means they change so that they can best perform the jobs for which they are created. Differentiation is away from the meristem. So you have the zone of elongation, the zone of differentiation, which are things you learned at high school. Okay, thank you.